So last week we got through the class averaging part of the tutorial. So building sets, generating reference-free unsupervised class averages. So we did that last time, but we didn't really go any further than that. So the next step would be this optional step of eliminating some of the bad particles. We have other strategies for eliminating bad particles now, which is more automated. Well, this can be pretty automated as well, but it's not a bad thing to understand what's going on with this step at least. So let's just take a look. So since last week, I went ahead and ran several different things. So I ran a couple of canonical refinements, and I ran refinement using the new refinement program. I ran the 2D refinement a couple of different ways. Sorry, the 2D class averaging a couple of different ways. Anyway, this gives us a variety of different things that we can look at while we're talking about the rest of the tutorial today. So I have both the sort of the canonical approach, but also some of the newer approaches which aren't fully documented in the tutorial yet. So let's just do E2 display. And we can take a look at my R2DB and R2DB01 folders. Now, if we look at R2DB, this was run with the full resolution particles. So if we look in here, we'll see that we have 256 by 256 particles in our class averages. And if we double click on that, we can see these particles. I'll shrink it down so we can see more of them at a time. And you can see it did a fairly nice job. Now, these particles are not filtered in any way. So if we look at the raw particles that are associated with these class averages, it can be a little difficult to see anything. So for example, if I double click on this, that'll show me all of the particles that went into that particular class average. And you can see that it's a little difficult to really see the particles there. I mean, you can tell that they're in the boxes, but it's not terrific. So it can actually be better when you're trying to do this sort of getting rid of bad particle thing, to actually use filtered particles for that process. So if I open up instead, so I reran the class averaging process, the reference-free class averaging process, using the downsampled particles, so one of the LP data sets. And if we look at those, you can see they're smaller. And they look similar in terms of contrast and that sort of thing. But if we double click on one of these, we can see a little bit more detail in the individual particles. And what you'll see is that a couple of these classes, like this, seem to have a lot of obvious ice contamination and stuff like that in them. Now, it's not a tremendous number of particles here, but they're definitely present. We can look at this one, and we can also see that there's a bunch of issues there. This one, again, there's a bunch of bad stuff in there, and it's kind of obvious because of sort of the really high contrast and the funny shapes. The funny shapes are really obvious, but if you see something like this, maybe that's actually a particle. It's just in a particular orientation where it looks kind of like that. That may or may not cause you problems. So if you want to go through the process of getting rid of bad particles or trying to reduce the number of bad particles in this data set, there's another program in the project manager. And this process is described in the tutorial. So if we look here, eliminate some of the bad particles. You're told to run this 2D analysis, mark bad particles by class. Right, so here under 2D analysis, mark bad particles by class, and that'll open up this interface where it'll show us all of the different class average sets that we've generated. This will also show us class averages that were generated as part of the 3D refinement process, which you could also do stuff with. But basically, this just shows you all the different sorts of class averages that were generated in the project. So if we click on this one, R2DB01, that's the reference-free, the unbiased, or the reference-free class averaging from the second run. If I single click on that, That will open up several windows, which will start out on top of each other, so you'll need to rearrange them. So it's got an excluded particles window. And just like everything else, once you've gone through this, once you quit the program, it should remember where you put all of these windows. So if you open up the same program again, it should put them in a sensible order on your screen. So here again are our class averages, and if I say, okay, well, I didn't like the particles in this class average, I can, sorry, and again, it resizes the windows the first time it displays them. It's an annoyance, but anyway. If I single click on it, it will show me the included particles. In other words, the particles it thought were good in the class average, and the excluded particles, the particles which weren't included when it generated the class average. If I double click, it'll put a little blue mark up in the corner. And then I can mark all of the data sets, all of the class averages that I think are bad. Maybe I don't like this one, obviously. These particles were automatically excluded, so it's kind of okay that they're there. And these particles look like they may be kind of okay, but it's a little bit of a low contrast mess. So maybe I'd want to get rid of that one too. Most of the rest of these are actually pretty nice looking. So once I've marked them, then I can say mark as bad. And then if I build new sets in the future and I tell it to exclude bad particles, then it'll make new sets which don't include any of the particles that I've marked in blue. And that's a progressive process. There's just one mark that you can put on each particle which says this particle is good or this particle is bad. So once you've marked the particle as bad, if I went back to this program again and picked some more and I marked them as bad again, that would just expand the list of bad particles. Or I could make a new set, and making a new set would make a set which contained only the particles that were marked. So if I did that, I would wind up with a set which contained only the bad particles. So if I want to get the good particles using the Make New Set button, I would hit the Invert button here, and that would invert the selection and select only everything except for the things that I had previously marked. And then I could say Make New Set, and then that would give me a new .lst file in the sets folder, which would contain the particles excluding the bad ones. So I'm not actually going to do that right now because I've already run refinements and that sort of thing. I just sort of explaining the logic of this program to you. This program can be used for a bunch of other things as well. The idea is that it'll do various operations based on class averages. Any questions about that before I move on? Okay. Again, that process is optional. You don't need to do it to get good results out of this out of the tutorial. It's potentially useful if you have data sets that have, for some reason I can't seem to resize my windows. That's weird. Oh, it's the smallest looking. Okay, that's fine. Okay, so once we've moved past that, this optionally marking bad particles, we move on to generating an initial model. Now, there are a couple of different strategies you can use to make an initial model. One strategy is you can look at your 2D class averages, and you can pick a handful of good particles from the 2D class, or good class averages from the set of 2D class averages, and then use those with the old style initial model generation program, Make Initial Model. Or there's a new program here called Stochastic Gradient Descent Initial Model Generation, which doesn't actually require class averaging at all. And in fact, it will often run better if you give it raw particles than it will if you give it class averages. So if we want to run this approach, first thing we need to do is go into our particle generation. I'll go ahead and use the downsampled class averages for this because it'll help the program run faster when it runs. Then I open up my set of class averages and I look at them, and I want to pick a few representative particles from all of the different views that are present here. So the way I do that is I middle click, and if you want to include most of the class averages and just exclude a few, you can use the del button, and you can delete the ones that you don't want, and then you can save everything that's left. Alternatively, if you just want to include a few, so if there are fewer that you want to keep than you want to get rid of, Then we use the sets interface. Okay, so I actually already made a selection here, cell, which was a previous selection that I did. So if I check this checkbox here, you can see these green marks appear in the upper corners of the particles that I picked. Okay, if we pick a set and we want to add a class add one of these class averages to the set, like say I wanted to include one of these things which could be an end view, I just single click on it and I can toggle it on and off. Okay, but first I have to select the set over here that I want to modify. Once I've got the set selected, then I can just toggle things on and off for that set. I can also show other sets at the same time, and you can see there's two different colors there now. And again, hopefully you are seeing numerical labels in the lower left-hand corner here. I haven't gotten around to fixing that on my Mac yet. Macs seem to have this particular problem. It doesn't seem to affect other programs, like when you do the 2D plots and you drag around, the fonts seem to work correctly there, but they're still broken here. So I don't know what's going on with the black boxes thing. I need to take some time and look at it. Anyway, once you have picked the subset of particles that you want, you hit the Save Set button, and then you can save them to an HDF file. Okay, 17 images appended to good2.hdf. So now if we close out of this and go back to the main folder, you'll see we have this file good2.hdf. That's when I did it earlier. And if we open it, you can see only the particles that I'd selected are shown here. Once you've got your good particles, then you can go over to this Make Initial Model box, and you can select the file name that you just produced, the file containing just the handful of good particles, and then fill in these few options here. Now, since I'm using particles here that were already downsampled, right? These particles are already 84 by 84 pixels, not the 256 by 256 that the original particles were. I don't need to use this shrink option here. If you used the 256 by 256 particles, then maybe you'd want to reduce those down by a factor of 3 or a factor of 4 or something, just to make this initial model generation run faster. And it'll run a lot faster if you reduce the particle size. Generally speaking, the scaling with box size in most of the operations in most single particle packages scales as, it's like the box size cubed or worse in many cases. So you can get a tremendous amount of advantage out if you downsample your data in terms of speed. Eventually, you're going to have to go back to the original data if you want to get the maximum quality out, but you can save that for sort of your final refinements. Anyway, so you can just set up your options, and when you're done, you should see that there's a folder called Initial Models. Okay. These other folders, init model 00 and 01, we're using the stochastic gradient descent. We'll talk about those in a minute. But you'll start with this initial models folder, and if we look inside there, you'll see a bunch of numbered files of several different types. So I ran this program 2 times. The first time I ran it, I used all of the class averages that came out of my second refinement run, the downsampled particles. And the second time I ran it, I used only the selected subset, the 17 that I decided were good. So what you see here, this model 0001, this is the first run of E2 initial model, and this is the first model that came out. So when it generates initial models, it doesn't just generate one, it generates however many you asked for. And then in theory, it sorts them in order. So the best one should be first and the worst one should be last. So I told it to generate 8. So this should be the worst one and this should be the best one. That's not always perfectly true. Its mechanism of judging whether it's got a good one or a bad one is based on similarity between the class average and projections of the 3D model. And there are cases where you can have very good agreement between the class averages and the 3D model, but still have a wrong structure, particularly if you have a small number of class averages. So I ran it a second time, and the second time I ran it, it only had 17 particles. If you look over here at the times, you can see all the times are about the same here, and that's because it doesn't write the output out until at the very end. So you can't really judge how long it took to run based on that. If you're interested in seeing how long a program took to run, there is a hidden file called .eman2log.txt, which has a log of all of the EMAN2 programs that you ran in the current folder, other than things like E2 display. Anything that generates output will produce this an entry in this EMAN2 log file. So we can do something like grep E2 initial model in EMAN2 log, and we can see a line for each time I ran that program. So you can see the first time I ran the program, I used classes sort 0, 0. So that was all of the class averages. Then the second time I ran the program, oops, sorry, sorry, this is the other initial model generation program. So I ran the other one first, and then I ran the new one. So the first time I ran I ran the old one. So the first time I ran the old one, I used all of the class averages. And then the second time I used the selected ones. The other thing you can see here is at the beginning of the line, it tells you when the program started running and when the program finished running. So you can see that it took 7 minutes to run the first time. And when I ran it with a smaller number of class averages, it only took about 2 minutes to run. And on top of that, I generated more class averages when I ran it the second time. So here I said tries 16, and here I said tries 8. So this one ostensibly would have taken twice as long to run. Anyway, so you can look at this file if you want to get any details about the history of the jobs that you've ran and stuff like that. In theory, you should be able to look at this log file and see the exact commands that you use to produce any particular file that you generated during refinement. So this serves as sort of a scientific log of what you've done in the project. Of course, it can't keep track of non-EMAN2 commands. Only EMAN2 commands count.
Why did you try 16 the second time? I would actually have thought that you would have done it the opposite when you have fewer.
Well, the more tries, the better chance that you're going to get a good class average. I probably should have tried 16 up here too, but since I was using a lot more class average, it was going to take longer to run, and I didn't want to sit around and wait for it. That wasn't.
Oh, sorry. Okay.
So here I was using a large number of class averages, which makes it run slower. So right. Since I was just doing it as an example, I thought I would run it with fewer. Of course, that means it's less likely to produce a good answer, but anyway.
Gotcha.
All right, so let's look at what we got. So initial models. So there are 4 different types of files that the initial model generator produces. This is the actual 3D map. This is the initial model that you would use if you decide it's good. This, where it says init, this is what it started with when it used its random generated refinement process. This is not something that you should ever use. I know it's got init in the name. That's the init for the initial model generation. This is the output of the initial model generation. Then it has this A particle file, which allows you to compare the projections that it used with the class averages. And then it has a projections file, which shows you projections of this in all different orientations. Now you can generate this sort of on the fly using other programs available in the system. So, but it's just there for convenience, basically. All right, so let's take a look at the model that we got, and we can see what it looks like. So that was model 1 as an output. So actually, I usually start by looking at the A particle file, because that gives you a better picture of whether things worked well or not. So the idea here is in the A particle file that you have alternating class averages and projections. So this is a class average, and then this is the projection of this corresponding to the orientation, corresponding to the orientation this class average was believed to be in. So if things went well, you should see this and this agreeing, perfectly to within the limits of noise. So you'll see here that some of them agree reasonably well, but others agree really poorly. Like, this sort of end-on view thing didn't come out with anything. This and this, not really the same, right? I mean, there's some agreement over here, but so this is not this is something that I would view as actually a pretty bad result. And this is what came out as our first, what should have been the best result from that trial of running with all of them. Okay, someone's annotating my screen. Could whoever did that clear their annotations? Or I'll do it.
It might have been me, but I don't know how I did that.
Okay.
Sorry.
You must have brought up the annotation interface and drawn. Anyway, all right. So let's take a look at the second one. Maybe the second one is better than the first one in this case, and we'll see that. No, again, it just didn't come up with very good answers. Now, this may be because I only told it to do 8 trials, and it just didn't wind up with a very good initial guess. It's a stochastic kind of process. So you probably you probably want to try 20 or 30 or something like that. The advice in the tutorial is you try running it once with 10, and if you don't get a good answer, then try it again. That can work sometimes. Anyway, let's see what happened when I selected a set of class averages. So that was the second run. So this is the first output from the second run, and look at the A particle file from that. So now there's fewer to compare because I only used 17 inputs. And oh, actually, look, that actually seems to have done a pretty decent job, right? All the class averages and projections here now actually seem to agree pretty nicely with each other. All right, let's take a look at number 2. And again, number 2 also seems to agree fairly nicely. Each class average and each projection seems to agree pretty well. So let's take a look at this 3D map. All right, doesn't actually look all that different than the one that we got out of the, right? So this is the one we got when the first run. This is the one we got from the second run. And I know that this particular class average is not really a very good one. Let's see what it looks like when it is good. So the second one we got out this time is actually a very nice initial model. So let's, oops, I lost my control panel. Where did it go? Oh, there it is. Okay, so this is the one I don't like. And this is the one that I do like. You can see it's a lot narrower. It almost looked like a 4-fold symmetry in the other one. And this one is sort of shorter in this direction. But that means it also has a projection which is sort of more elliptical. So if we look at the projections, so let's look at the projections from the one that I didn't like. So this is showing us projections in all different orientations. So all of our particles should look something like one of these projections. Oh, sorry, that's 0, 0, 0, 8. That's the wrong one. That's the worst one from the previous round. This one. There we go. So all of our projections should look, or class averages should look something like one of these. Now, the thing you'll notice is that this looks very nice. Some of these look very nice. But then when we get up here, we see all these kinds of artifacts and stuff in the projections. And what this means is we didn't really give it a complete set of orientations. So the subset of particles that I picked didn't have very complete coverage of the asymmetric unit. It was missing some orientations, and the way it arranged things, it came up with something which is just a little funny. So let's take a look at the second one, which I think looks better to my eye. Okay, and now we can see that one of the, some of the views are actually sort of look like that circular thing that we discarded as bad. Now, it's a little more elliptical than some of the averages that we were seeing. So there was some ice contamination mixed in there. But to my eye, this looks like a more self-consistent set. And if we look at the 3D map, it is more correct. Now, how can you judge that? You can't necessarily. If you don't know anything about what your particle looks like, it can be very difficult to judge. If you look at the A particle file and you find significant disagreements, if you have a class average which looks good and you have a corresponding projection which doesn't look like the class average in some significant way, even in 1 or 2 cases, that's usually a bad sign. Now, if the class average that you're comparing to doesn't look very good, if it looks really noisy and like it jumbled a bunch of stuff together, then you could look at the particles that made up that class average, and you could say, oh, was it really including particles that all looked similar to each other or not? You can ask more detailed questions. Probably I didn't try it, but probably if I used this one that I didn't like as much as an initial model, it would probably still converge to the correct final 3D structure eventually. But not necessarily. You can come up with initial models which are all very self-consistent. So this one was very self-consistent. All the class averages and projections matched each other, which means that unless there's enough particles in those other orientations to kind of fix things, you could wind up in a situation where it'll just iterate and get stuck in that local minimum forever. So there are some gotchas you can run into here once you start doing the 3D refinement in theory. Okay, so this was using the old initial model generator. I would actually suggest, generally speaking, using the new initial model generator, and it mentions it in the tutorial, even if it doesn't take you step by step through using it, mostly because it's pretty easy. So if we just pick stochastic gradient descent here, and we pick one of the raw particle sets, so not particles, sorry, sets. So if we pick, you could do it on the full resolution, but again, it'll run faster if we use one of the low pass filtered ones. So I would use LP12 or LP5 here. And just give it the raw data set, fill in the symmetry, and you can just leave the other parameters pretty much as they are. You probably want to update the number of threads to match the number of threads available on your machine. Other than that, though, the options are the defaults are generally pretty good. You can change the number of tries if you want. Anyway, so I ran that, and I ran that twice. Unlike the other program, which generates sequentially numbered files in the same folder for each iteration, this new program follows the more conventional approach of each time you run the program, it makes a new folder. So if we look at an init model 00, we'll see the 10 models that I generated that run, and there are only 2 files for each one here. There's the A particle file again, which lets you compare individual particles with the corresponding projection, and then there's the 3D structure. Now this file, if you're using raw particles rather than class averages, isn't as useful. Now when I ran this the first time, I ran it with class averages just to show you, oops, sorry, that was the wrong window I was using. That's fine. Let me put that back to what it was before. Well, it doesn't matter, I'm just gonna cancel. Let's get rid of that. Okay. There, init model 0. So I ran this the first time, I used class averages as inputs. And if we look at the A particle file, you can see some of them agree kind of, but it didn't really do a terrific job. This program is stochastic, meaning it only uses a subset of the particles in each iteration. So if you're going to use this program with class averages, you don't want to just give it 64 class averages. You want to give it 1,000 class averages or 500 class averages or something like that if you want it to work well. And you can use class averages with it. It's just that you want to generate a lot of them. Alternatively, you can just throw the raw data at it. You can just skip the 2D class averaging process entirely if you want to. I don't recommend normally just skipping it entirely. Usually you want to do the 2D class averaging just to get a feel for what the different orientations of your particle actually look like, given that the raw particles are often pretty noisy looking. So even if I'm not going to use it for the initial model, I'll usually still generate these class averages. But for making the initial model, you can actually just use the raw particles with this. And most of the time, it'll work pretty nicely. Now, if we look at this, you can see it's going to show us a handful of comparisons between individual particles and class averages, but the particles are really noisy. So you can say, oh yeah, look there and that, actually that does look pretty nice. And that, actually that does look pretty nice. But the particles are pretty faint, a little bit hard to see, even though they've been filtered a bit. So you may not get as much out of the A particle file in this case, but we can look at the 3D map. And if we look at the 3D map here, sorry, this is an overlap again. I'm just adding more 3D maps to this window as we go. So here's the 3D map that we got out from the initial model generator using all the particles as raw data. And you can see, it's not too bad. It's maybe a little bit wider than it should be, but it's almost certainly fine for purposes of refinement. It has all the features that it needs to have, and this is pretty typical. We have very, very rarely seen this new initial model generator, the stochastic gradient descent one, fail to produce something which would converge to the right answer when given raw particles as input. So nowadays, this is actually the approach that I use myself. Just use the stochastic gradient descent if you need an initial model. If you're working in a system that you're already familiar, you can just use an existing structure that you have, which is similar to something as an initial model. That's fine too. But if you're starting on a brand new project, this is very nice. All right. Any questions about initial model generation? Okay, I guess I give a really good explanation. Hopefully everyone got at least one good initial model out. And if you didn't get a good initial model out and you refined it anyway, which happens sometimes during the tutorial, I would say usually about 75% of people manage to get good class averages and run the old initial model generator and get a good initial model and produce a nice structure at the end. Then the other 25% make some errors when they're picking their class averages, like they use all of them, and they wind up with a funny initial model generation because we're trying to push the speed when we're doing the tutorials, and they wind up with a refinement which doesn't work right. And those are useful outliers to show other people what happens when things go wrong. My refinements didn't go wrong, so I won't have a lot of great examples of that to talk about today, but I did the 3D refinements in two different ways. So if we look at the tutorial doesn't talk about the new 3D refinement strategy yet. This tutorial was last updated for EMAN 2.31, and the new refinement program kind of first existed in the 2.91 release from last year, and has been improved since that time. So we haven't added it to tutorial yet, even though it's probably the thing that you want to use moving forward if you're going to do single particle refinements anyway. So refine 00 and 01 are kind of following the canonical refinement strategy, which is outlined in the tutorial here. We'll skip through all this. Yeah, 3D refinement. I have a slightly better computer than the one we assume that people have in the workshop, so I maybe ran a little bit longer with a little bit more aggressive parameters in these two. So this is actually targeted to be a fairly high resolution refinement, and this was targeted to be sort of an intermediate refinement. But anyway, I ran two refinements here, and I also ran one of the new style refinements. So we can take a look at the results of all of these different things. So if we look at the first refinement, we can see a bunch of files in the folder.
Real quick, could you explain just like the significance of how you came up with the names, like R2, like what's the, I guess, refine 3D?
You want to know the names of the folders?
I guess that's in the refine 2D. Is that where it comes
so the
from?
names of the folders. I think the init model and init initial models, that's pretty straightforward.
Right.
R2DB, that stands for refine 2D, and the B stands for bispectrum. In the 2D analysis, there's a reference-free class averaging, which is the old style class averager, which works really nicely, and sometimes you might still need to use, but takes a lot longer to run because it's iterative. And then the new bispectrum based ones, the B stands for bispectrum.
Gotcha.
Then refine, that should be pretty straightforward. That's 3D refinement, and R3D, refine. Refine 3D. It's the new program. No good reason.
No.
3D and refine.
Cool. Yeah. All right. Thanks.
Okay. Right. So refine 00. If we look in this folder, we'll see a bunch of different files here. I will point out that there is a place in the wiki where it describes what all the different files mean. It explains some of them in the tutorial. The main things you would normally want to look at in this folder are the class averages. Those are classes class is 0, 1. So this is the first iteration. I ran 5 iterations in this folder. So these are the class averages. The FSC curves. So we have FSC curves generated for unmasked, masked, and masked titer versions of the refinement. And I would point out that even the tight mask here isn't quite as tight as the mask Reliant typically uses for its post-processing strategy. There were just limits to how far I was willing to take that tight masking strategy for pushing resolution, measured resolution. Sometimes you might want to look at the projections. So the projections should match the class averages. So sometimes it can be useful to sort of switch back and forth between projections and class averages. And then the 3D files, these are the actual outputs. So 3D05.hdf, that should be our final result. So we can always start by just looking at the final result and see what it looks like. And again, I've still got all the other ones that I've done in here. Let's raise the isosurface threshold a little bit. And you can see that this actually refined to pretty decent resolution. A lot of those details you see there, those are actually real. Now there is a little noise outside the structure, but this quick refinement I ran, as I said, was a little bit more targeting a little bit better resolution than the refinement that it tells you to start with in the tutorial. Actually, let's take a look at that. Let me close all these windows just so we're sort of starting from scratch here. Open a browser again. The other thing you can look at in this refine folder, there's this refine parameters.json file. I showed you here what the .eman2 log file, which logs all the commands. So you can find the command that was used to run this refinement in that eman2 log file as well. But if you did something like just save the refine00 folder as a part of an archive or something, this file also contains all the parameters which were used for the refinement. So if you hit info here, you can go into this JSON browser. Note that this info button does different things for different types of files. And if we look here, command line, that will show the command line that was used for this refinement. So you can see I used the full resolution particles. I didn't use the downsampled particles here when I ran this. And I used init model 01. So I used the results of the second run of the new stochastic gradient descent initial model generator. I set a target resolution of 8 angstroms, and that limits things. So if I say a target resolution of 8 angstroms and I get an FSC curve which shows that it refined to 5 angstroms or something like that, I can't really trust it. I need to run, and that means I need to do another run where I set the target resolution to at least the value that it's getting to. Speed equals 5 was my concession to actually running it kind of faster than usual. So if you say speed equals 5, it will sample things less, and it'll run a lot faster. It'll run maybe 5 times faster or something like that. It's not speed equals 5 doesn't mean 5 times faster, but it's sort of the default medium value. If you want to run your final full resolution refinement, you usually use speed equals 1. Or sometimes 2 or 3 is enough. Speed equals 5 will run a really fast job. Symmetry D2, this top hat option, that's explained in the tutorial in detail, but that's the reason why the map looks kind of noisy, because I use the top hat equals global. It sharpens things and doesn't do anything to sort of low pass filter the noise in places where it's bad. Top hat equals local would have produced a nicer looking structure, a smoother looking structure with less noise outside. It takes a little longer to run. 5 iterations. I think all these other options are pretty straightforward. Note that I specified parallel thread equals 8 and threads equals 8 as required, and also as explained in the tutorial. All right, so we can look at the class averages and see how they look. And these progress from sort of the view along the z-axis here, and then they sort of spin around gradually. Actually, if I open this in the 2D view and I use my mouse, you can sort of see how the class averages are generated. You can see how it's rotating as I go through the class averages sequentially by holding the down arrow down. If I middle click, I could also use the N slider oops, the N slider. Oh, hey, I'm having that bug again. All right, that's another Mac specific thing. Right. So, so you can look at the class averages, and if you want, you can compare the class averages to the projections. So if I scroll down here, as soon as I double click on projection 01, it'll switch all of these with the corresponding projections. And ideally, they should all look the same. Now, obviously, if you have some class averages which are really noisy, they're still going to look really noisy. There's nothing to do about it. But in general, you should see good agreement between the class averages and the projections, particularly once we get to the last iteration. So here's the projections from the last iteration. And here are the class averages from the last iteration. And you can see even at high resolution, they agree pretty nicely now. All right. So if we want, we can open the 3D models in sequence. So 01, 02, 03, 04, 05. So they're all loaded together now. Now I'd like to adjust the ISO surface so it's not showing us quite so much noise. There is an option, I think if I pick all objects and utilities, no, not all objects, right? Tree view, oh, there we go, miscellaneous under all objects. You can pick a single value for the ISO surface threshold and it'll adjust all of them to be the same. So we can look at just one of these, the last one, and we can pick a good ISO surface threshold for it like that, maybe. So about 3, then I can go back to all objects here. I can type 3. Now they're all adjusted. And now if I go through them sequentially, you can see there's the first iteration, second iteration, third iteration, fourth. So you can see even the first iteration already started with a pretty decent high resolution kind of looking map. If we want to evaluate that a little bit, we can look at the FSC curves. So here we've got FSC. So let's look at the masked tight FSC curves. That's usually the sort of the final output. So that was the first iteration, second iteration, third iteration, fourth iteration, fifth iteration. So you can see it converged, and it converged to a resolution of ostensibly something around 4.8 angstroms. Now, when I told it to refine, I only told it to target 8 angstroms. So that means that this result is not valid. The first iteration, it had already gotten past 8 angstrom resolution. So really, if I was going to use the fully sampled, unfiltered particles, I could have just run sort of my full final refinement on this with a high target resolution and probably even skipped this because I started with a good initial model. So if we look at the second refinement that I ran, the one that was targeting higher resolution, we can look at the refinement parameters in this one really quickly. Sorry, command line. And I told it to start from refine 00. So that's going to use the same general, it's going to use the final structure that came out of refine 00 and the same data set. So it's for doing continuations where you want to change some of the parameters. Here I set the target resolution to 3, the speed to 1. And other than that, I kept pretty much all the options the same. I probably should have changed top hat to local here, but I didn't. So we're going to have to suffer with the noisy looking map. But anyway, we got a pretty decent result out of this anyway. All right, so let's look at the Fourier, the FSC curves from the second run, and we'll see. Oh, look, it started there. So ostensibly that got to about 3.7, 3.8 angstrom resolution. It probably would have been very slightly better if I had used the top hat equals local, because it would have locally filtered out some of the noise and it wouldn't have contributed as much to the FSC curve. So local resolution can actually have an impact on your measured resolution, but not a profound impact, just a minor impact. So let's take a look at that 3D map, the final 3D map that we got out from that second round. And again, we'll have to adjust the ISO surface threshold on this final map. Sorry, there we go. Around 3, 4, something like that. As we rotate it around, you can see it's a little noisy. It would have been nice if we'd filtered some of that out. But we can compare that to that. So you can see it has more features inside it, and that sort of thing. Anyway, it refined to a pretty nice structure, and we could compute an FSC between that structure and the known structure for this data set if we wanted to, and I'm sure we would get a very nice number, although I didn't actually do that at this point. Okay, great. What about this new refinement program I was talking about? So you can see a lot of features in here, right? You can see this, you can see the strands. There's a beta sheet right in here. You can sort of resolve the strands in the beta sheet a bit. But the structure still looks a little noisy and kind of falling apart a little bit. But overall, it's at a good resolution. If we just filtered it a little better, it would look very nice. Actually, we can just do a little bit of a global filter on it. Let's go ahead and we'll launch filter tool on this, and we can play with the filter. Turn off the slice, and I'm going to say filter low pass Gauss. Cut off frequency. Let's filter it to say 4 angstroms. Okay, so now that's been filtered to 4 angstrom resolution. You can see it got rid of a lot of that funny noise outside, and just visibly, it's a lot easier to see a lot of those features now. If I adjust the isosurface threshold a little bit, we'll pretty much get rid of all that noise. And we've gotten rid of a few of the details which are real, but it's a lot more pleasant to look at this way. Anyway, you could play with that filter threshold a little bit. You could filter it to say, maybe 0.2. So that's a 5 angstrom filter, and see what it would look like, sort of 5 angstrom low pass filter. Or we could filter it less. Now, if we filter it to less than the resolution that we've, to beyond the resolution that we've achieved, you'll notice it's still doing a filter. It's still getting rid of some of the noise. So this is where it's important to point out that Gaussian low pass filters are sequential. I mean, meaning if you low pass filter something to 4 angstroms and then you low pass filter that volume to 4 angstroms again, it's not going to stay the same. It's going to be filtered some more, and you're going to wind up with an effective filter of something like 6 angstroms instead of 4 angstroms if you do that. So the filters are sequential in that way. Now, if you do a top hat filter is where you go out to some resolution and you just do a sharp cutoff to 0, then that isn't true. Then if you filter to 4 angstroms and you filter to 4 angstroms again, the top hat filter will be exactly the same after both of them, because it's basically just throwing everything away at high resolution. It's not doing anything at low resolution at all. The downside of doing top hat filters is they also produce ringing artifacts. So if we do, let's see, 0.5 here. Sorry, not 0.5, that's 0.2, so 5 angstroms. So 5 angstrom Gaussian low pass filter. We can compare that to a low pass top hat filter. And you can see some of the nasty stuff that it does to the structure. And if we turn the slice back on, you can actually see what's going on a little bit better, right? So that's, I changed the units when I changed the filter. That was a more aggressive filter. There we go. So this is the top hat filter. You can see all these ripples coming off the structure and that sort of thing when we do the top hat filter at 5 angstroms. If we do a Gaussian filter, and again, I've got to adjust the resolution. You can see that it's constrained locally, right? I mean, it chops off the edge of the mask. You don't see any of these funny ripples running out. It's a lot smoother in general when you use the Gaussian filter. All right. So anyway, we got some fairly nice structures out of these quick refinements that I did. approaching the sort of resolution that I said you could get if you did, you know, a nice final refinement in the third round over here. I also ran the new refinement program. The new refinement program, you look at the command line there. This uses CMD instead of command line. It's there wasn't a reason for that. Muiwan just didn't think about it before he did it. Anyway, that's what it is in CMD. We can look at the command line there, and you can see this command line. Again, it was using the full resolution particles. We went back. We were using the same initial model that we used in the other program. 8 threads, symmetry. This isn't a target resolution here. In this program, the resolution is the starting resolution. So we're starting at about, telling it to start by low pass filtering at 18 angstroms or phase randomizing at 18 angstroms. Keep says that you want to throw away 10% of the data, and then we're going to run for 8 iterations. This program typically requires more iterations to converge. The amount of time that it takes to run is pretty comparable to doing the high resolution refinements over there. We can look at that. It can take longer, it can take less time. It depends on the particular project, but it generally tends to produce nicer answers. So let's take a look at the results of this. 3D08. And oh, hang on, let's see. Oh, sorry, I've got them all loaded here again. There we go. So you can look at this. Now you can see these beta strands there. They're very nicely resolved. There's a nice alpha helix there. You can see the helix inside. There's another beta sheet in there. As you can see, this actually just visually, it just looks a lot cleaner. And if we look at the FSC evaluation on the final, actually, we can go through all of them here. 0, 1, first iteration, second iteration, third iteration, fourth, fifth, sixth, seventh, eighth. So you can see it wasn't making much progress once it got past about the 5th iteration here in terms of resolution. But what resolution did we get to? Oh, this got to 3.4, which is better than the 3.8 that we got before. So how long did it take to run? So you can see this finished at about, the last iteration came out at 8:45. The first iteration came out at about 10 p.m. the previous night. We can see, so here you see about an hour and a half per iteration here, something like that. If we go and look at the second refinement I ran with the speed 1 option, so the slower refinement there, you can see each of these iterations, the FSC, that's easier to see. Each of these iterations took longer, took 4 hours to run. Okay, the other refinement I ran, refine 0, 0, the first one with speed 5, that one took about less than an hour, right? So a little less than an hour to run with speed 5. But you can see we started with just the initial model and we just immediately got to the final answer. We didn't have to run multiple programs. It just refined very nicely to the final answer. So this is, needless to say, this is the approach that we're moving to as we approach EMAN2 here. And you can use it now if you have a current snapshot of EMAN2. I can't guarantee you it's going to work perfectly in every case. This is still sort of a beta program, but from my experience, it works very nicely most of the time. This is not using deep learning or anything like that. It is taking advantage of some of the newer optimization strategies available in SciPy. So it's hybrid between EMAN2 and tools available in SciPy. Anyway, it works pretty nicely and is definitely worth a try on your own projects. And it's pretty easy to use. It's not part of the GUI yet. So if you look in here under 3D refinement, I don't think we have the new refinement there. in the subtomogram averaging, which we're going to talk about next week, we do have the new subtomogram averaging programs all integrated into GUI, but we haven't done it yet for the single particle.
So I had a question regarding that point.
Good.
So I did refine this data set after generating a nice initial model, and I used your single map refinement, and I set the parameters as is listed on the tutorial, but unfortunately I got a indexing error.
Indexing error.
Yes, and afterwards my terminal crashed, so maybe I'll have to have a look again and try to replicate that process again. But I remember getting an indexing error, and I wasn't able to proceed from the first or the only 3D refinement, rather.
Can you share your screen?
My EMAN is on another laptop. This laptop doesn't support Ubuntu, but if you like, I can send you the screenshots later.
Okay. Well, the idea was that we talk about problems that people encountered kind of interactively, right? So,
Right.
but I can look at it. I usually, if you encounter some error, you probably didn't follow something in the tutorial right. I mean, the tutorial has been tested over and over again in many laptops and many iterations. So usually it means you failed to follow some instruction. If I see exactly what it is, I can probably tell you where you went wrong.
Yes.
But beyond that, does anyone else have any problems that they had or successes or whatever that they want to discuss? I pretty much just talked and showed you what I got running various ways. How many people actually got what they were supposed to get? If you didn't get what you were supposed to get when you went through the tutorial, do you know why you didn't get what you were supposed to get? If you don't know why you didn't get what you were supposed to get, then now would be a great time to talk about it.
I have it's not a question related to your last point so much, but when you do the initial models, there seemed to be a lot of variation in the initial models. So I don't know. Can you talk a little bit more how you finally decide on one? Or I don't know. I don't have a very good question.
Usually, the first one is going to be the best one.
Okay.
With either of the programs, I would say that if you run the new stochastic gradient descent program on the raw particles, generally speaking, the first thing you get out is going to be fine. Usually, if it's not fine, it'll be pretty obvious that it doesn't really look like a structure. So if you give it particles which are highly variable, a lot of heterogeneity, then that could cause problems, right? I mean, it may not succeed in producing a single good initial model there. Those are the situations where running the 2D class averaging is key. Not so much that you can pick class averages to use for the initial model generation, although you could, but more from the perspective that you can actually see what sort of heterogeneity you have among your data. Maybe when you pick particles, you actually pick two different particles. You know, say you were picking ribosomes and you pick whole ribosomes, but you also picked a whole bunch of 50S subunits alone, right? And then you mix those together. Now, in that situation, actually will still generally refine to the right answer, but then you'll wind up with a final 3D map, which has a stronger 50S than the whole, you know, the 50S side of the part of final reconstruction may be stronger than the other side because it's missing in a bunch of the particles or something like that. I'm just saying that there are a lot of situations with heterogeneity and variability and stuff like that where that can cause issues. In this particular case, with the beta galactosidase, it turns out that the key problem, I'll go ahead and share my screen again if no one else has anything that they want to show. The biggest problem that you run into with this data set is the lack of end views. That's the reason why it actually goes wrong. If we look at the class averages again, Okay, so here are the class averages, and now let me go to the 3D refinement. And we'll look at the projections from the end. All right. Oh, let me turn off the label. All right. So what you'll see is there are views that look kind of like this. Maybe when we get all the way to the end here. So you can see there's a lot of projections generated by the time that we're getting there. There are these sort of end-on views like this, which basically look like a bright ring with a little bit of a elliptical area around the outside of it, but it looks sort of primarily like a bright ring. And what happens in this data set is if there are a significant number of these ice contamination particles, those views get mixed in with these ice contamination particles. So this is actually, you can sort of see the central lobe and you can see this little bit off to the side. This is actually an important view. And if you were looking at this data set just from scratch, if this is the first time you'd ever looked at this data, you would probably, double click on one of these and conclude that, oh, that's primarily ice contamination. I just want to get rid of all of that. And you might wind up throwing away the particles that were in that orientation. And without that orientation, it's actually kind of hard for it to come up with an initial model because it's important. Now, if you get some of the intermediate tilts, so if you get orientations like, this with this sort of bright lobe here and that sort of thing, if you get enough of those in there, that can sometimes fix the problem. But anyway, for beta galactosides, you have this sort of pervasive issue, which is that one of the views of the particle has a tendency to look a lot like ice contamination. Now, if you have a good data set that doesn't have any ice contamination, that can help. And I'll also point out, oh, actually, I didn't do that, which is the second strategy for, I didn't show it to you. The second strategy for looking at bad particles. So if we look at the tutorial again, after running the refinement, it talks about, oh, where is it? Sorry, I skipped over it. Here we go. Identifying bad particles in the data set. So I used the deep learning particle picker when I did the particle picking for this. So if you use the predefined particles, you get about 5,000 particles. And a pretty good chunk of the 5,000 particles are actually ice contamination in the data set that was provided when they published the paper. When you use the deep learning picker, it does a much better job of getting rid of those ice contamination particles. So I got about 10,000 particles, and only maybe 500 to 1,000 of them are bad in some way, probably actually even fewer than that. So there's the lobe, I guess you'd say, is kind of smaller. Anyway, so if you run this program, which basically compares each particle to its corresponding projection in a variety of different ways, it produces this, there we go, particle fsc00.txt file. If we look at that, we can compare various aspects of the particle to each other. So this particle, this data file has 19 different parameters. So it's got one row per particle. So there were 10,733 particles in the data set, and then it has 19 different values that it computed for each particle. So we can look at any of those 19 different values. So the first value is the Fourier shell correlation at low resolution between the particle and the corresponding projection for each particle. The second column is sort of one notch higher resolution, and then the third column is one notch higher resolution, and then the fourth column is another notch higher resolution. I think it describes that here somewhere. Yeah, here. So the first four columns so 30 to 100 angstrom resolution, 15 to 30, 8 to 15, and 4 to 8. So we can plot on the x-axis. Now we're plotting the very low resolution features, and then on the y-axis we're plotting the somewhat higher next notch higher. If we do the next one, we can still see we're getting a little discrimination on the y-axis, even at sort of intermediate resolution. And then now we're not getting very much at all. So if we plot, say, 2 versus 3, we're not seeing much of a distribution. So this is the second highest resolution band versus the highest resolution band. Now the first 4 columns are from the second to last iteration in the refinement folder. Then the next set of 4 columns are from the last iteration. So you can compare the second to last iteration with the last iteration and see how self-consistent things are. So we can compare column 0 to column 4. And if everything were exactly the same, if the refinement had perfectly converged and nothing was changing from one iteration to the next, you would see a perfect diagonal line here. So all of the stuff out here is probably garbage because it didn't include very well. Now, if I mouse over some of these things, it can actually show me the particles which correspond. So each time I move, so you can see here, this is actually a little blob of ice contamination here. There was some ice contamination. Anyway, if you mouse over these things, they should look like really nice particles. Anyway, the conventional thing that we look at here is visualize, 4 versus 5 or 0 versus 1, and you can see these 2 lobes. And generally, this is good. Stuff up here is good, and the stuff down here is bad. So if you just run that program, it will automatically split the data up. And the way it does it is you can do it manually here. If we say classification, and we tell it to use, I don't think it actually uses all axes. We can just tell it to use all, and we say number of segments 3. Then we can wind up with, oops, I don't know, let me get, oh, there we go. Now I got 3. All right, let's not use all axes. Let's just use 0, 1, and 4 and 5, and we'll do that because it doesn't use them all when it does it normally. So you can see why it classifies the data, and turn off the blue ones. So you can see the red are sort of the bad particles, and the green are the sort of good particles. I'm not sure why I didn't get any yellow that time. All right, hang on. I managed to confuse things somehow. Let me just do this one more time really quick, and we'll wrap things up. Let's try it again. Again, you don't normally have to do this manually. It does it automatically for you when you run the program, and it'll produce, if we look in sets here, it will produce PF00 bad and PF00 good. So you can see it found 749 particles that it thought were bad and 9,984 which it thought were good. So you could then run another refinement on this data set. This is a small enough number of bad particles, and they're mostly going to get automatically excluded anyway. It's probably not going to have any really major impact here. But if you go through the tutorial and you use the particle data set that they provided, the ones they had picked, it's actually more like 15 or 20% of the particles that they had picked are actually not very good. So you're like 5,000 and 1,000, something like that. So then it actually can make a big difference if you get rid of them. Anyway, it does this automatically, and the way it does it is by doing what I was showing you here. It picks a specific set of columns. Oops, right. It picks a specific set of columns that it knows are useful. Splits it into 3 groups, and then it keeps the 2 groups of the 3 which it knows are the best. Yeah, once again, I only got 2 out. I don't know what's going on with that. It's safe.
Why did you choose 0, 1, 4, and 5 for the
there we go.
axes?
It worked that time. So 0 and 1 are the lower resolution FSC values, which tend to be the most reliable. And 4 and 5 are also the lower resolution, but they're from the last iteration. So I want to look for things that are sort of self-consistent between the second to last iteration and the last iteration. And I want to look at things, and I want to look at sort of the lower resolution FSCs, which are sort of the most predictive of things that are bad. So I'm trying to get rid of things like ice contamination or
Right.
particles that are just really weak overall. And they'll tend to have a really low correlation at low resolution.
Gotcha.
So the actual thing the program uses isn't just 0, 1, 4, 5. It actually uses some of the other columns that I didn't talk about as well. If you look at that text file, where is it? There we go, that one. If we look at that text file itself, oh, sorry, it strips the labels off the top. Let's do that. I killed it, so it told me I'd killed it. At the top, it'll actually, it has a set of labels which tells you what it's got. So here it gives you the resolution ranges for the first iteration and the second iteration that it looked at. Then there's an RMSD between the two, which is basically the agreement, how close they are to that slope 1 line in the corresponding plot. Then we've got something comparing the intermediate resolution to the low resolution. Then we've got orientation parameters here, that sort of thing. So when it does the splitting between good and bad, I believe it uses maybe the first 3 resolution bands for both iterations. And I think it uses this RMSD parameter, which is sort of how well the 2 iterations agreed with each other. Maybe it uses that too. I'd have to look at the program. Anyway, the automatic thing is automatic selection of the bad particles is generally pretty good. Okay, I think we've gotten through everything I was planning to cover for the single particle refinement section of this. So the goal would be then for next week, we would start going through the subtomogram averaging tutorial. So if you go to the tutorials page in the web page, there's now a unified subtomogram averaging tutorial. If you have done this previously and say, oh, I've done this before, I don't need to look at it again, the tutorials have changed pretty substantially. The tutorial now makes use of some of the newer programs that were developed over the last couple of years, and there's no big tutorial and small tutorial anymore. Now there's one tutorial which includes both the big and small variants of the data set. So even if you did it in the past, I would suggest, if you want to get a lot out of it, to go through the tutorial one more time. It shouldn't really take more than a half a day to do sometime. So that tutorial will do a subtomogram average of the ribosome, and if you really go through it fully with the big data set, you can get to sub-nanometer resolution on the ribosome from just a handful of particles. The other nice thing about that is after we finish the subtomogram averaging, then we're going to go and we're going to take a look at the new Gaussian mixture model tool. And the Gaussian mixture model tool supports subtomogram averaging now. And actually, the ribosome data set that we use in the tutorial is actually a really nice example for that. So I'm planning to run the GMM tool on the results of the subtomogram averaging tutorial. So if you haven't done it, then when you get to that the GMM stage at the end there, you won't be able to follow along as well. Okay, any questions about anything before we go? All right, well, hope everyone got something out of this today. And feel free to email me with questions if you were too shy to ask while you were on.